Welcome to the Fundamentals of Image Quantification lecture. This is the introductory lecture to the Fiji for Quantification workshop. Hopefully you're here because you want to learn how to turn this nice fluorescence microscope image into some kind of quantitative data. In order to do this, there are a number of steps. First of all, we need to look at the planning side before we get to the experimental side. And really the first thing that we need to do is to actually think about the endpoint quantification that we're wanting to do. That will determine all of the steps that we have to go through from there. Once we've determined what needs to be measured, that will inform us in terms of how to acquire the data and which microscope to use. That will also inform us in terms of how we should prepare the sample. Once we have the sample, we can acquire the data. Again, this may change depending on what the quantification is that you're required in terms of your instrument settings, acquisition settings. And then finally, we can work on actually measuring that data. One thing that we would suggest strongly is to run through each of these steps as a pilot ahead of doing the final analysis, because what you'll find is you'll often need to go through multiple rounds of optimization for each of these steps to get the entire workflow working properly before then running your full-scale analysis. If you are a bump user or from the University of Melbourne and you find any problems optimizing any of these steps, then please don't hesitate to reach out and consult with us, and we can try and advise you as best possible. Okay, today's talk is going to be set out roughly on the kind of general workflow of a bioimage analysis experiment. So hopefully you've taken some very high quality microscopy images which you want to analyze. Normally the first thing we'll do before getting to analysis is to perform some form of pre-processing just to make sure the data is suitable for analysis. We'll then go through the image and identify the regions that we want to analyze. Very often this is through object segmentation. Before coming to the measurement and data analysis size of the objects that we've identified, and then finally going on and reporting or putting it in a paper or a publication. So we'll go through each of these steps in turn. We're not going to touch the image acquisition. You can find other content on that on our website. So we'll start with pre-processing. Now, these are some examples of pre-processing that you may want to perform on your microscope images. Again, it depends on how the data has been acquired. The first one is filtering. So these are very useful for reducing the effect of noise, or even high resolution effects within images if your data has been taken with too high a resolution. Two commonly used examples of this are the median filter. In this case, it looks at pixel X and looks at the pixels directly surrounding pixel X and takes the median intensity of these pixels and assigns it to pixel X. The second example shown here is Gaussian blur, where we apply a blurring algorithm, a convolution, to the image. The nice thing here is we can select a strength to be applied, which can really affect the strength of the blur within the image. This slide will take you through some of the effects of these on real data. So this is a very noisy microscope image on the top here, and on the bottom we have the effect of performing a normal intensity threshold, which we'll cover over the next few slides. This shows you the effect of the median filter and the Gaussian filter. One thing to point out here is the median filter nicely maintains the edges of the structure, which allows for some more detailed segmentation, whereas the Gaussian blur is applying a smoothing, which doesn't maintain the edges of the structure quite so much, and therefore has a downstream effect in terms of the regions that we can select within the image. One other thing that's also available, especially within Fiji, so there's a noise to void plugin, which is a pre-trained deep learning plugin, model, which will can be applied to noisy images, and again, we'll clean that data up for downstream analysis. Again, the preference is to not create data which is quite so noisy, but if you have, then there are ways to clean that up. The next example is background correction. So you can see in this example here, at the center of the image is nicely illuminated, but towards the edges of the image, there's a little bit less intensity. So it's not evenly illuminated across the full field of view. There is a tool called rolling ball, which will allow you to even out that illumination across the image. This is an example of flat field correction. So again, here the illumination, this time in transmitted light, is not so even across the entire field of view. Again, with a reference image, we can correct for that in this example. If we have some striping here, effects that were taken during acquisition, again, we can use a bandpass filter to clean these up. And then also we have a cross-section of brain here, it looks like, which we've tiled across to take higher resolution across the entire section. And we've corrected for the shading correction within this. I would argue for the majority of these that you should be able to clean these up at the time of acquisition. That again is preferred to having to do it in post-acquisition. One thing to watch out for, especially in time-lapse acquisitions, is photo bleaching. In this example, you can see this object, which is bleaching over multiple acquisitions, which would make a traditional threshold approach to identify this object very, very difficult. So there's a bleach correction tool. This shows the intensity over time of this object. And with applying the bleach correction algorithm, you can see this has cleaned up that data much, much more. And you could use a traditional threshold approach to actually identify those structures over time. This is an example of that applied to some live cell data with the bleaching on the left and the corrected image on the right-hand side. Next thing we might want to do is correct the alignment of the sample. So it may have drifted in multiple dimensions during the acquisition of the data. There are also effects from the optics of the microscope. So for example, chromatic shifts through the objectives. So lights from different wavelengths not behaving the same way. We can measure this with fluorescent beads and use that to correct the alignment. Again, especially important if you're doing things like co-localization analysis. And then finally, stitching of 3D volumes together to make a much, much larger image. We can use plugins such as Big Stitcher within Fiji to perform this as well. The last pre-processing that we often suggest to people is deconvolution. So if we were to look at a point of light as it goes through a microscope, what we see at the other end is what we call a convolved image. So there is some blurring and some effects from the optics of the microscope. So it's not the same coming out as it is going in. What deconvolution does is it applies a mathematical algorithm to try and return the signal back to its original state. Again, we can measure this with beads and measure the mathematical function and try and help return that back. That will allow you to look at these structures here, where we can see that these are really elongated. Signal to noise isn't necessarily that fantastic, but now we've gone through and deconvolved, then we can see we've actually increased the resolution and the signal to noise on those samples. And again, there's this example down here, deconvolved image on the left-hand side, deconvolved image on the right-hand side. And again, signal to noise is much, much improved on the deconvolved data, which will allow you to identify structures for quantification much, much more easily. Okay, so once we've pre-processed the data, we can go through and start to identify the objects that we want to quantify and potentially segment them out individual components within the image. So very often what we're doing in this case is taking a pixel segmentation approach where we're looking at the intensity within an image and saying, okay, this is the region that we want to image. Using a threshold approach, we can create what's called a binary mask. Basically, we're telling the software we only want to analyze within this white region here. There are multiple ways of doing that, and we'll cover some of those in the Fiji for Quantification workshop. So as I mentioned, conventional thresholding, machine learning approaches, using deep learning models, and lots of other ways as well. Once we've got this binary mask, then there are lots of things that we can do to that. So we can perform watershed functions to try and replicate as close to the structures as possible. We can do things like ultimate eroded points to try and find the center of the objects as well, and we'll cover lots more in the coming slides. It all comes back to our fluorescence image, and this is a digital image, and the image is made up of pixels which have intensity values associated with them. So the intensity range is determined by something called the bit depth. So here we've got an 8-bit image, that's 2 to the power of 8, so that gives you 256 grayscale values from 0 out to 255. If you had a 12-bit image, that would go up to 4,095 grayscale values. 16-bit goes up to 65,000 grayscale values. So what we're using very often, as I mentioned, was the intensity within the image to define this is my signal versus this is the background. Some of the other things we can do within the intensity range is to do things like find maxima. And again, we'll use this in one of the hands-on activities. So in this case, we're looking for local maxima. Works really nicely identifying nuclei. And we can separate out the spaces between objects using segmented particles, using a watershed function to define the gaps between these objects that we've identified. Similarly, we can use edge detection where we find the edges of fluorescence signals, and that again will allow us to identify some objects for downstream analysis. As I say, mostly we were using conventional segmentation. We'll do this quite a bit during the hands-on activities. So again, this is our intensity histogram. So we've got low intensities down here. So at the 0 range, they come up black in the image, and high intensities are in the bright green up here to 255 grayscale values. Again, the important thing is you don't saturate, so you're not hitting this maximum value. That allows you to really work well within your image data. So what are we doing when we're applying a traditional threshold? Essentially, we're saying at this point within the image histogram, anything above that point, we're going to define as being our signal, and anything below that point, we're going to define as being our background. That, as I say, we will use to produce a binary mask for downstream analysis. But the question is, where should you put that threshold limit? Often we suggest that you use a negative control or look at the intensities in a background area to try and define what that threshold should be. And really, to be objective, you should try and apply the same threshold to all of your images if possible, but that's not always possible. So very often you'll find real images have very different background levels. You know, your background may change over time within the sample if you're doing live cell experiments. So how should we put thresholds on these 3 different image histograms? So Fiji has a way of doing this. So we have auto threshold algorithms, which again we'll go through in the hands-on activities. This uses a number of published ways of determining what a threshold should be. And Fiji will go through and test all of these for you. And each of those has its own specific mathematical way of determining what the threshold value should be. So there's auto threshold methods. So you can input your fluorescence image and it will output what the mask would look like for each of those different methods. There's also an autolocal threshold, which we'll also go through in the hands-on activities, which uses a rolling ball filter to correct for any background differences. And again, that will go through published methods to try and show you what's the best thresholds to apply. Once you've selected one that you think works best to represent the data that you want to highlight, then you can use that going forward for each of your images. Okay, so that's traditional threshold, and that works very nicely on fluorescence images normally. Sometimes, though, you're not always working on samples that are the best. So sometimes conventional thresholding will struggle on things like bright fields, electron microscopy, or color-based images, where the signal doesn't work quite so nicely as standard fluorescence images. So in this case, we are going to use machine learning, where we can manually annotate different parts of the image to define them as different regions. And within Fiji, there are a couple of ways we can do this. So there is a trainable Weka way of doing this, and there's also something called LabKits, and we'll show you how to use LabKit as part of the hands-on activities. So again, really nicely interactive, very easy to use, no coding or programming experience required. If you did want to get a little bit deeper, if you pardon the pun, into segmentation, then there are lots of deep learning models available as well, which have been pre-trained on different data sets. So there's UNet here, which is trained on the bright field images. There's STARDIST, which is very useful for identifying nuclei and cells, and we'll go through STARDIST as one of the hands-on activities later on. And there's also CellPose as well, which is again pre-trained on lots of different cells, different cell shapes, and works fantastically well at segmenting out different objects, if that's of interest to you. Here, to develop them, they need a really large amount of manually annotated data and probably some pretty good programming skills to produce those models to start with. The nice thing is, as I say, there are some pre-trained models available, which you can use within softwares like Fiji or in Python as well. Okay, all of those, what we're aiming to do is convert this fluorescence image into this binary mask. And I've mentioned this already. Essentially, what the binary mask is an image with two different grayscale values. So it's got the zero intensities or a maximum intensity. And again, all we're doing is telling the software we only want to quantify within the white regions within the image. I already mentioned earlier on a little bit about binary imaging processing. So again, we can identify those structures using what we call a watershed function. We can start to separate those out. We can go through what's called an erode step, where we can make those slightly smaller, or through a dilate step to make them slightly larger. And all these are useful for us to try and clean up our binary images down the track. We can skeletonize, so again, bring this down to a very, very simple model of cell shape. If our binary mask has holes in the middle of it, we can use the fill holes function to fill that in. And I think I already showed this. We can also use ultimate eroded point. So if you keep eroding until you find the final pixel, that in theory is the center point of your object. And one of the activities that we'll be doing is to use a combination of binary images and also the grayscale images to generate specific masks of individual cells. And again, we can use this to quantify on a per cell basis out of an image So we can get a lot more information than just average stats across the image. We can get a per object stat as well. Okay, I mentioned about cleaning up some of our binary mass. Some of the other things that are useful in this space are some of the mathematical functions or something called Boolean logic, which we can use if we combine two images. And again, this is something we'll play around with as part of the activities later on. As mentioned, useful for combining binary masks, removing objects, or highlighting different objects. So we'll go through the image arithmetic first. This is an example of an add function. So what we're doing is we're taking image 1 here on the left and image 2 on the right, and we're basically going pixel by pixel and adding the intensity from image 1 to image 2. Okay, so in this case, if we add this together with this, our final result is that the plus and the minus symbols have doubled in intensity, whereas the multiply and divide symbols should be the same as they are on the left-hand side. If we subtract, so in this case, the plus and minus symbols should disappear. We're subtracting one from the other, that becomes 0. And here we're subtracting the multiply and divide symbols from 0. So again, they should remain, whereas we lose the other two symbols. When we come to multiply, it gets a little bit more complicated. So here, this appears in both images. So we're multiplying the intensities here. And down here, we're multiplying by 0. So we should get 0 here. And this has multiplied up. And you'll notice we've gone from an 8-bit scale up to a 32-bit scale to be able to contain the intensity values for the plus and minus symbols. Here for divide, this also gets a little bit complicated. So this is what the result looks like. So if we divide this by this, we should get 1. If we divide this by 0, in this case, this turns into infinity. So this is again a 32-bit scale. The reason we can't see the plus and the minus symbols is because we're on a 32-bit scale, and these have an intensity value of 1. We just can't visualize that within the image, but the intensity value of 1 is there for both the plus and the minus symbols. So hopefully that makes sense. And again, we'll use that in the hands-on activities later on. Next thing we'll move to, and we'll use the same examples, are the Boolean logic. So in this case, we've got logical AND function. So what we're looking for is objects which are found in both images. So if we take image 1 and image 2, then only the plus and minus symbols are in both of the images. So we only maintain those. If we look at the logical OR, then this is an object which is in one image or the other image, therefore both images. And therefore we have all 4 symbols. If we look at the logical XOR, which is exclusive OR, then we're looking for something that's in one image or the other image, but not in both images. And in this case, we highlight the multiply and divide symbols here. I've already mentioned some of these binary options, although they do get a little bit more complicated from this point. One of the first things we'll do at the start of the quantification workshop is to make sure our binary options settings are correct. So in this case, when we hit the dilate button, we're adding pixels onto our object. And in this case, we're adding 1 line of pixels once to our objects. If we hit erode, then we're removing 1 line of pixels once from our objects as well. We can combine these. So there's a function called open, which is an erosion followed by dilation. And this smooths the objects and removes isolated pixels. And that's highlighted down here. So we've got a little object here that maybe we don't want to include in our analysis, and we can get rid of that just by going through an open function. Similarly, we can go through close, which is a dilation followed by an erosion. So again, that smooths objects and fills in holes. So there's an example here where maybe these two objects shouldn't be separated. And using the close function, we were able to join those back together again. Okay, once we've identified the objects that we want to analyze, the final thing is what we can measure and how we can analyze that data. So what we're going to go through is the set measurements function within Fiji. So these are all of the quantitative readouts that you can perform within Fiji. And we'll go through and explain what each of those are in turn. So to perform a measure task, we go to the analyze measure or control M or command M if you're on a Mac to actually run the measurements. To bring up this set measurements, again, similarly, you can go to analyze set measurements, tick the relevant ones, untick the ones that you're not interested in. In terms of what we can read out, so one of the first ones here is area. So that's the area of selection. So if you have a threshold there, that's going to read out just the area that you've selected within the threshold. The key here is you have to select the limit to threshold button. That area will be read out in whatever the set units are. So if your image isn't calibrated, it will be in pixels. If your image is calibrated in microns, then it would be in microns squared. We can also read out area fraction, and we'll do this in a couple of the activities. That is a percentage of the thresholded pixels in the total area of the image. So we're going to use this for calculating cell confluency as one of the activities. The next important thing that we can read out is intensity. So again, there are a number of things we can read out here. So something called integrated density will give you two readouts. So that's either the sum of the intensity units in the selection, or it's the mean gray values multiplied by the area. We can also read out the mean gray value, which is the average of the intensity units within the selection. Similarly, we can read out the minimum gray values within the region and the max gray values within the region as well. We can read out something called the modal gray value, which is the most commonly occurring gray value. So that's the peak here of the intensity graph. We can read out the median, which is the midpoint of the intensity distribution within the selection. And we can also read out the standard deviation as well. The next set of readouts are shape descriptors. So we can look at how circular an object is. So a perfect circle would have a circularity of 1, and there's an equation there showing you how that's worked out. We can look at aspect ratios. So we'll take our particle, fit it with an ellipse, and we can look at the major and minor axes of the ellipse. We can look at the roundness, which is the inverse aspect ratio. Again, the equation is there in terms of how that's worked out. And then we can read out the solidity of an object as well. So that's the area divided by the convex area. So for example, this circle or this ellipsoid shape would have very, very high solidity. If you look at the star shape, then that would have a much, much lower solidity. So the way I think of the convex area is if you took a rubber band and wrapped it around your object, the footprint of that rubber band would give you the convex area. So here the star shape has much lower solidity than this circle, for example. We can also read out location. So first thing listed here is the center of mass. So that's a brightness weighted position. We can read out the center point of a selection. So there's the centroid. This is the example of the center of mass. So because more of the intensity is to the top right of the object, the center of mass is shifted to the top right. We can measure the perimeter, so the length of the boundary around the outside of an object. And we can also measure something called Fairey's diameter, which is the longest distance between 2 points on the boundary of an object. mentioned already about ellipses fitting and bounding boxes. So bounding rectangles up here fits the smallest rectangle into your selection. We can also fit an ellipsoid. And similarly, we can read out the position within a stack. So whether it's times time or channels or a Z stack, that can be read out as well. Some of the final ones that we don't tend to use very often is skewness. So that's how symmetrical the distribution is within an object. And kurtosis looks at the flatness in XY, whether that's Gaussian or flatter or more peaked than normal. But as I say, we don't tend to use these two very often. One tool we use quite a bit, especially when we're trying to do things like counting cells or counting objects, is the Analyze Particles tool. So this is the dialog box for Analyze Particles. This gives us a few options up the top here. This allows us to filter out objects within our image. So we can filter based on size and we can filter based on circularity. The show function here allows you to generate a new image after you've done your processing. And again, we'll show you in the next couple of slides what those different options are. These are them listed down here. Some of the other options we have are display a results table, clear an old results table, or summarize a results table. We can add the particles to something called the region of interest manager, and we'll cover that quite a bit in the hands-on components. We can exclude any objects which are touching the edges. Again, very important if your readout is area, that the entirety of the object is within the image. We can include any holes within our analysis. And again, the help button there is very, very helpful if you want to know any more about any of these functions. So first thing we'll do in this slide is go through some of the Analyze Particle filtering steps, these two based on size and circularity. So this is a demo image available within Fiji called Demo Blobs. And you can see they've got lots of different shapes. This is a threshold that's been applied to those and created a binary mask. In this case, we're selecting the exclude on edges, and you can see anything that's touched the edge of the image is excluded from the analysis. As I mentioned, that's very important if your readout is area or shape, that you have the entirety of the object within your image. When we select the include holes option, you can see that any holes that are identified within our objects are filled in for the analysis. In this case, we are filtering based on size and also circularity. So here we're only interested in objects which are between 0 and 50 pixels squared and are fairly circular, so have a circularity of above 0.5. Therefore, this identifies the small circular objects within the image and gets rid of any of the other larger objects. In this example, we're not filtering based on size, but we are filtering based on circularity. So anything with the circularity below 0.5 identifies these globular and elongated structures. You can further refine that. So between 0.3 and 0.5 gives you the more globular structures. Below 0.3 gives you just these elongated structures. And then again, you can come in, filter based on size, so anything larger than 50 pixels squared and fairly circular, so circularity larger than 0.5. So the greater than 50 pixels squared gets rid of these smaller structures, but we maintain these more kind of globular circular blobs. Hopefully you can see that would be useful in terms of identifying or removing different objects within your regions that you've selected. Next, we'll go through the show options. So again, as I say, this is a way of using Analyze Particles to create a new image. So outlines here draws outlines around the objects with a number associated with each of them. If you select bare outlines, then this produces just the outlines without the number. You can create a mask, and we will do this as part of the hands-on activities for further downstream quantification. You can get it to show you what ellipsoids were selected to enclose each of the objects. We have count masks here, where each object has a slightly different intensity value. We've got overlay outlines, which is outlines overlaid over your raw data. And then we have overlay masks, which again is a mask overlaid over the raw data as well. So that is probably enough from us just to get you started to think about the process and the workflow for bioimage analysis. If you still have questions, then feel free to either reach out to us, go to our website, and you can go through, there's a more in-depth introduction to bioimage analysis video as part of our Fundamentals of Fluorescence Microscopy series. I would also highlight this Image SC forum. That is a central forum for lots of the open source data analysis packages, of which Fiji is one. If you do encounter any problems with Fiji, that's a very good place to go to. Very often people have encountered these issues before and have posted the questions and have had answers in terms of how to fix it. Thank you.